Oscar is not able to join, so I will start and end this month's lecture. I'm very happy to welcome you all to this October lecture. It's a pleasure to welcome James Wason from the UK via Zoom. I had the pleasure to teach with James, I think 2 years ago, before the pandemic, in Wengen, and I'm looking forward to teach again with him, hopefully in a few months. I'm still quite confident that it will happen. James,
may I jump in? It's close to 2 years. You know,
it's close to 3 years. Okay.
No, 2 years. January 2020. So we heard the first sort of troubling news from China, but we still believed that it's all fine, but it wasn't fine at the end. Okay.
Yeah, you see that's psychology. It's also you forget about the times. Anyways, so it's a pleasure to introduce James to you before I will hand over. James is a full-blown, a true statistician, I'd say. Who studied statistical sciences at the University of Cambridge and then did a PhD there at the MRC Biostatistics unit. At that time, I think about genome-wide association studies and statistical issues around that. But nowadays, he's much more into clinical trial designs and adaptive and complex trial designs, which he will also talk about today. And he's now, since 4 years, professor for biostatistics at Newcastle University. The topic for today is about complex trial designs and innovative trial designs. And this is, I think, a topic that is of interest not only for trialists, but also especially for epidemiologists, because some of those innovative trial designs are used a lot for public health aspects, for public health interventions. For example, the just now started vaccine trial that is sponsored by the WHO, where CTU Bern is also involved, is a platform design. So there is several vaccines to be tested in one single trial. So you see, it's of relevance, I think, for all of the people in the audience, the topic, and I'm very glad to hand over to James. James, the stage is yours.
Thank you very much, Sven. Really nice to be able to talk to you all today. I'm sorry I can't be in Bern in person. That would be very nice, but maybe one day soon. So yes, I'm going to talk about innovative trial designs and what might future trials look like. I mean, to some extent, this is all already happening from the COVID situation and such like, but I will talk more about these innovative designs, including master protocols. I'll go into a bit more detail about basket and umbrella trials. You might have come across these in oncology, and I've got a big interest in how we can use them outside of oncology. I've got a few examples that I'm involved with in Newcastle. Then I'll talk about platform trials, which have been widely used in the COVID situation, so you may be familiar with them. And I wanted to finish by talking about a project that we've just finished in the UK about supporting adaptive trials and how to appropriately resource them. Sorry,
quickly, and you will do that all in 45 minutes?
Oh, I'll try. I might need to really rush through the last few slides. We'll
that sounds like a long list of
Thank you. I'll do my best to get through all of that. Yeah. Anyway, so I'm sure we're all familiar with randomized controlled trials where we're randomizing participants between a new experimental arm and a control arm, which might be a placebo, measuring outcomes, and then at the end analyzing difference between the two arms. And this is a gold standard way to show that a new arm is beneficial compared to an existing one. But they are also very expensive, and this has been contributing to the cost of evaluating drugs, which is now at 2.6 billion per approved drug, and it's going up fairly quickly. So innovative designs can really help through reducing the average cost of a trial, for avoiding doing underpowered trials, so wasting resources, and related to these things, combining what might be several different trials into one, using what's called a master protocol, or a platform trial being one type of them. So several types of master protocols exist, and I've got pictures of a few here. I'm going to go over these in more detail, so don't worry if you can't see them too well, but we have platform trials, we have basket trials, we have umbrella trials, and these all have the same property of combining what would have been separate trials into one. And each has got different advantages and possibly disadvantages, and may be suitable in different settings. So I'm going to start with basket and umbrella trials, and I would point out that this is almost exclusively used in oncology settings where treatments targeting particular tumour mutations are being tested. And a basket trial here is testing one of these targeted drugs for patients who are positive for a certain tumour mutation, but each part of the basket trial is a different location of the tumour. So you might have breast cancer, you might have lung cancer, etc. But all of the patients would be positive for a particular type of mutation that the drug targets. Umbrella in oncology, at least, is similar, but it's testing multiple targeted drugs, and it's generally in patients with a particular tumour type, like breast cancer, and each of the drugs is targeted at a different tumour mutation. So this is what it might look like with a basket trial and an umbrella trial. So an umbrella trial, we've got the different patients who are positive for different biomarkers or tumour mutations in oncology would receive different treatments, whereas in the basket trial, it's just one treatment being tested in different locations of the tumour.
So what would be the control groups in these trials?
Oh,
going a bit quickly for me.
Good question. I mean, in oncology, in basket trials especially, it's quite common to not have a control group. But I'll be talking a bit more about outside of oncology where that's not the case. In umbrella trials, there are often control groups, but not in that diagram I showed. But you might have, say, these patients randomized between control and this treatment, and you might have these patients randomized between control and that treatment. So you could have single arm or randomized comparisons.
Thank you.
Basket trials can be randomized as well in oncology, but it's much more common that they're single arm. But I'm going to show a few examples outside of oncology, which look a bit different. But just to give an overview of the benefits. So one is operational benefits of running several trials within the same protocol. This should be more efficient than what you'd need to run all of them as separate trials. There's also statistical benefits where we can apply innovative statistical methods, and I'll say more about that later on. And they might also be popular with patients, especially umbrella trials where the treatments are really targeted at the specific type of patient. And the idea, hopefully, is that those treatments would be highly effective for the patients who've got particular tumour mutations. So I think these types of trials should be used much more widely, and they are starting to be used outside of oncology, but it's still 90% plus in oncology. I think a basket trial could be of use whenever there's distinct related diseases that either share mechanisms or share symptoms such that a common treatment is likely to be beneficial to multiple ones. I'll show you an example of a trial in a sec. Umbrella trials, I mean, could be used anywhere where there's multiple treatments available that are likely to work differently for different subgroups of patients. So let's have a look at a trial that we're running in Newcastle. So this is just started recruiting, had a lot of delays due to COVID, but I believe first few patients were recruited last month, which is great. So this is, I'd class as a basket trial. It's called OAKS. It's a common treatment, which is at a symptom that is common to multiple different conditions. So we have primary biliary cholangitis, which is an autoimmune liver disease, which surprisingly perhaps causes issues with cognition as well. We have actually two stages of PBC, so early stage and late stage. But we also have Parkinson's disease, which is quite different, but has that symptom in common as well with the cognitive impairment. So each of these trials is essentially a separate randomized trial with a placebo and the same treatment, and will be analysed separately. So we will do an analysis of each trial separately. But since we've got a lot in common here, we can combine the results as well and do an overarching Bayesian analysis, which I'll say a bit more about later. This is an example of where there might be mechanisms and symptoms in common, such that a common drug could be useful. Here's another example. So this trial is not yet started. We are hoping to, we've applied for funding and unfortunately weren't successful, but we hope to gather additional evidence to justify it. And this is in Sjogren's syndrome, which is a very heterogeneous disease that has many different symptoms in different types of patients. And research at Newcastle University has developed a stratifying tool that can divide patients into different groups based on symptoms and other information. And so we have, in total, there's 4 different types of patients, but one of them is a low symptom burden group, and they're not experiencing many symptoms, so won't likely agree to be part of the trial. So we're concentrating on the other 3 groups. And each of these has a predominant symptom that the trial, if it happens, will then stratify and allocate a treatment that's targeted at that symptom. So we have rituximab, we have hydrochloroquine, and a device, a vagus nerve stimulation device. And these are all targeted for the predominant symptom that the patient has. So in a sense, this is a bit like an umbrella trial. We've got one disease, but we've got different subgroups, and we've got different treatments with each one, although we've got the same control group. Here's another one that looks quite different.
And you also have the same outcome measurements?
Well, actually, that's a good question. In each trial, there will be a different primary outcome measurement, but there's a common secondary outcome measurement that could be used in a statistical analysis. So okay, yes, good question. There are elements in common with secondary outcomes, but not with the primary outcome.
Shall we collect the questions for the discussion later on? So just write them in the chat. I will pick them up at the end. Huh?
So the third example is a trial being led in Cambridge, but I'm involved with, and I designed the trial and I'm leading the statistical aspects. And it's in vasculitis, which is a rare autoimmune disease. It's actually a collection of diseases, as I'll show on the next slide. But the diseases have symptoms in common that inflammation causes damage to blood vessels. And there's several different biological therapies that could be useful. And we wish to do this trial, and it's just started recently. It should have started about 2 years ago, but got delayed due to COVID. And it's going to be using an interesting design that is kind of like a crossover trial. So there's 4 different treatments, the 3 biological therapies and placebo, and patients are randomized to a random sequence of them, like a crossover trial. But in this case, there's an endpoint which is time in remission, and patients will be kept on a treatment whilst they're in remission until they have a relapse. And once they have a relapse and then there's a washout period, they'll be moved on to the next treatment in the sequence. So if each patient is followed up for 2 years as we plan, we would imagine there'll be between 2 and 4 treatments that they receive in that time, depending on how beneficial they are. And this is quite a rare disease, and that type of design was thought to be very appealing to patients because they'd be kept on treatments that they benefit from. So I could give a talk just on that trial because it's quite interesting in many ways, but I just wanted to focus on the umbrella aspect of it, which is that vasculitis is actually a collection of very rare diseases. And so you've got all of the ones that are included in BioVas here. And the primary analysis, which will be in 140 patients, that's the sample size, is powered for a pooled analysis of all of these together. But we also have less good, but some power to detect highly effective biological therapies for individual rarer conditions. And we're working with Thomas Yaki, who you might know of, who is going to be doing a Bayesian elicitation exercise, so that even the very rarest of these diseases we can get a Bayesian analysis for, although it's going to be using informative prior information, which of course isn't as reliable as trial data. So all of these are really examples of what I would call basket or umbrella trials, but they look quite different to ones in oncology. And they each have some elements in common. So in OAKS, the intervention, the control, and the outcome is in common. In the umbrella Sjogren's trial, well, one of the important secondary outcomes is in common, but not necessarily the primary ones, and the control is in common. And in BioVas, the interventions are control and the outcomes are in common. So all of these have potential for applying advanced statistical analysis approaches, which I'm going to very briefly talk about and give you a paper if you're interested in more. And I'm going to be focused more on a basket setting. So this might be what type of results we get from a trial like OAKS, where we've got the 3 different trials and we estimate the treatment effects and get some sort of interval. So how do we analyze that type of data? Well, we've got a separate answer for each one, so we could just use that, just use the data from the respective trial to estimate the treatment effect, or we could assume they all have the same effect and pool them together. So these would make separate assumptions. So the separate analysis assumes that there's a distinct treatment effect in each group, in each trial. Complete pooling assumes all trials have identical treatment effects, and that's what we're trying to estimate. And therefore they have advantages and disadvantages. So this approach, if we're right and they do actually have the same effect, then this is going to be the most efficient way of testing for an overall treatment effect. But it's going to give misleading answers if that assumption is wrong. Whereas this first one, well, it's going to be very robust to there being different treatment effects, but it's also going to have the least efficiency. And so there's alternative approaches that are a compromise between the 2 extreme approaches here, perhaps, which allow borrowing of information across the different trials. So you may well have come across Bayesian hierarchical models, and this is a common way to borrow information. And a Bayesian hierarchical model would assume that the treatment effects themselves come from an underlying distribution. So perhaps a normal distribution with an underlying mean and variance. And small values of the variance here would mean that the treatment effects are very similar and would mean that the information is borrowed across trials. Whereas large values of this variance mean the treatment effects can be very different and less information is borrowed. And so there's various ways of using Bayesian hierarchical models and doing this type of thing, but they all assume in some way that the treatment effects come from some underlying common distribution, which may not be the case. In the OAKS example, we have 2 trials which are in PBC and 1 in Parkinson's disease. And so we might feel more comfortable sharing information between the PBC trials than we do between PBC and Parkinson's disease. So that motivated considering some alternative approaches. And actually, over the last 2 or 3 years, there's been quite a few different approaches proposed. This is just one of them. So this uses a Bayesian discrepancy approach. I'm not going to go into that much detail. But basically, it allows borrowing information between each pair of trials, depending on how similar they are, and also some prior distributions. So the idea is that if it looked a bit like this, so let's add a new trial so it's a bit clearer. But if we had this setting where we actually have 2 trials that have similar treatment effects and another 2 trials that have similar treatment effects but very different, then this is going to be a setting where a hierarchical model is going to not do very well, because it's unlikely that this comes from some underlying normal distribution. But the Bayesian discrepancy approach would allow you to borrow information between these 2 when analysing them, but not borrow any information from these 2. And similar, when analysing any of these 2, borrow information from the other one, but not from the other 2. So it allows a more kind of flexible approach to borrowing of information between different trials. And that's the approach we're going to be applying in OAKS as the secondary analysis. So if you're interested, then there's a lot more details in this paper, and I would encourage you to have a look. Okay, so I'm going to move on to platform trials now. And a platform trial, I think I've got a picture. Yeah, so it looks like this, where we've got potentially multiple experimental arms compared to a common control group. Now, there are some settings where you wouldn't have a control group, but most of the platform trials do have a control group. So we've got multiple arms. We've got possibly an adaptive design that allows, as we gather information during the trial, it allows us to drop arms that appear less promising when compared to the control group. And also, that would be illustrated here. So we might do an interim analysis here, and this arm 2 is stopped early because it's not showing much benefit. Just to say, this is over time as we recruit patients. And then we also have the opportunity to add in new arms as they become available. So in this case, we've added in this 3rd experimental arm here. So I wanted to quickly say about these 3 different elements and why they're good. First of all, the multiple experimental arms. Well, when we have multiple experimental arms available in the same condition, then combining them into a single multi-arm trial provides a big gain in efficiency when you've got a shared control group compared to doing separate 2-arm randomized trials, mostly because of the shared control group, meaning that you need fewer patients allocated to control. But there's also other advantages. So I think not many patients who enroll on a trial do so with the objective of getting the control arm, and a multi-arm trial will generally have a lower chance of them getting the control arm and a higher chance of getting one of the experimental arms. That's not always true. Maybe as the number of arms increases, it means that there's more potential that a patient might object to one of them. So there's a bit of a balancing act there. But it also means that we can compare the arms, the experimental arms, more easily compared to conducting separate trials, although I know there's some meta-analysis experts here who would have good ways of doing that for separate trials. Disadvantages, I mean, even though the increased efficiency, a multi-arm trial is going to be bigger and more expensive than doing a single 2-arm trial. It also raises the controversy of multiplicity, which I won't go into, but I've done some work on. And related to this point of comparing intervention arms, this might not be a desirable thing if the interventions are funded by different industry partners. They might not want their drug to be compared to another company's drug. So that's multiple experimental arms, a multi-arm trial. Next, an adaptive design. So an adaptive design gives you the opportunity to make changes to the trial as you gather information on patient outcomes. but doing so in a statistically robust way that controls the statistical characteristics of a trial, like type 1 error rate and power. And there's lots of different types of adaptive trial. We're mostly thinking of ones which allow early stopping of less promising arms. And in that case, it's going to result in shorter trials, or at least more arms being evaluable with the same number of patients. Perhaps better for patients enrolled on the trial because they've got less chance of getting ineffective arms, and more information on better arms. So this is a MAMS trial, so a multi-arm, multi-stage trial that I'm involved with. That's in Newcastle. Like many others I've mentioned, it's just started recently, but it was meant to start quite a while ago, but got delayed from COVID. And it's comparing two drugs versus control for orthostatic hypertension. I won't go into too much detail, but it's got an outcome that's measured at 6 months, which is something I'll come back to later. And the initial randomization is equal, but after 200 patients have been randomized, we'll do an interim analysis, which will use the 3 and 6 month data on the outcome to compare arms B and C versus control, and decide whether we should continue both arms or just one arm, or stop the trial if neither of them are showing any benefit. So that type of thing is what an adaptive design allows. All of that's done whilst knowing the type 1 error rate is controlled and the power is as planned. So advantages, I think, pretty clear advantages with the early stopping of ineffective interventions. Disadvantages, so we do need to do interim analyses quickly and to a high standard to make the right decision. I'll come back to that later if I have time. And we also need to observe relatively quickly compared to the length of recruitment. Again, I'm going to come back to that later. Lastly, the last element of a platform trial is the opportunity to add in new arms. And I think I heard Max Palmer use this analogy, and Max has really done much of the work on implementing platform trials in practice. He used the analogy of a steam train for a trial, where as you set up a trial and get it going, it starts slowly, but you open more sites and the recruitment rate increases. And then at some point you reach the maximum recruitment rate, the maximum momentum, and you answer your question, you've reached the required number of patients, and you shut everything down. And that seems like a bit of a waste, when you could add in new treatments as they become available and carry that momentum over to the new arms that are being assessed. So STAMP trial, which Max Palmer was instrumental in, did exactly that. So it's been going for a very long time. Of course, this is from 2000, I think, so it was forecasting ahead a bit. But it started off with 6 different arms, one of them being a control. And it took quite a while, as you can see, to answer the question. So going from starting recruitment of patients to getting the final analysis was taking quite a while. But as the trial went on and new arms were added in, you can see the time taken to get an answer was much shorter. So there's a real argument that this approach is beneficial to increasing the time taken to reducing the time taken to evaluate treatments. And this has also got the multiple arms versus control. It's also got the adaptive design. So this is a really good example of a platform trial that's had a big benefit. So when do these types of trials provide benefit? Well, you have to have the multiple interventions. You have to have good collaboration in the area, such that you can agree a common master protocol. You need a sufficiently high number of potential patients to recruit. You need commitment to ongoing funding for the underlying platform, as well as funding for new arms that get added. And for the adaptive part I've mentioned already, you need an outcome that's observed relatively quickly, or some informative short-term outcome. And that leads me on to thinking about, what elements here in the platform trial are the most beneficial and which are beneficial in some cases, but not all. So if we go back to these three things, we've got the multiple experimental arms, the adaptive design, the opportunity to add in new arms. Well, I think 1 and 3 here, I think they're always going to add efficiency as long as you're allowed to do them. So of course, there's maybe some obstacles from regulators to adding in new arms, although they have been very much more open to that in recent years. But property 2, the adaptive design, which is ironic because I've done a lot of work on adaptive designs and an advocate for them in general, but in my view, it's the least useful part of the platform trial, at least in some situations. And back in 2019, along with some collaborators, I actually had gotten a bit fed up of people asking if I could help with an adaptive trial where they had an outcome that took 5 years to measure and they were planning to recruit all patients within 2 years. That's clearly a case where an adaptive design is not going to be beneficial, because by the time you've got the outcome on the first patient that you've recruited, you've already recruited everyone and treated them. So there's no way an adaptive design is going to be useful there. But there's extremes here, of course. So that's one extreme, and the other extreme is a very short-term outcome where an adaptive design is always going to be beneficial. But where between the two, so 1 year outcome and 2 year recruitment period, that's probably not going to be an adaptive design is probably not going to be very useful, because by the time a decent number of patients have gotten a 1 year outcome, you're probably going to be near the moment. I've been very interested in looking at when an adaptive design actually does provide benefit, and we have a PhD student in Newcastle who's doing some interesting work on this. But I did wonder whether this is the same for these type of ongoing platform trials, or whether it's different. And I'm also interested in whether it's the same in different disease areas. So platform trials have almost exclusively been used in oncology and COVID, although there have been some uses outside of these two. But these two have the common property that you can't generally re-randomize participants who are enrolled in an arm that was dropped from the trial. So if you have one question I get asked all the time when I talk about adaptive trials and trials that allow dropping of arms is, if you drop an arm for it not being beneficial to patients, what do you do with patients who are on that arm? And the answer to it is, well, it really depends. I mean, if the patient's no longer eligible for the trial because they've had treatment and they're not meeting the inclusion criteria anymore, then there's not really much you can do other than perhaps withdraw them from the treatment if they're not benefiting from it and follow them up. But in more chronic relapsing remitting diseases, which I'm interested in, like rheumatoid arthritis, for example, you could have patients that during the trial, because it's a chronic condition, they might still be eligible if they'd come along at that point to the trial. So if they were on an arm that was dropped, could they be kind of re-randomized between the remaining arms? And that's something, when talking to patients with these conditions, they feel is very beneficial and should be done if there's no reason not to. But to my knowledge, it's not currently done, and there might be some statistical implications that are a barrier. So I'm interested in looking at that. And if you did that, then it would really also improve the efficiency provided by adaptive trials, because when you drop an arm, all of those patients are now available for re-recruitment to the trial. And there's also the other part of the equation. So the benefit of adaptive designs is the cost of doing these innovative trials. So that's the last thing I talk about. I think I'm doing okay for time. I've got 10 minutes or so, which should be enough. I wanted to talk about the Costing Adaptive Trials project, which is something that we have been doing in the UK. So in 2019, a group of us started and got funding for doing the Costing Adaptive Trials project, and some of the funding was used for a nice logo. an advantage of the acronym being CAT. And this was funded by the NIHR in the UK. And so a lot of us were kind of working a lot in adaptive trials, and we'd seen that they weren't being necessarily used as much as they should be. And one of the barriers that had been identified was funding them. So our objectives were to investigate this and to actually investigate how much extra funding, how much extra staffing resources and other costs are needed to support adaptive designs compared to non-adaptive designs through doing a mock costing exercise. Because it's hard to compare it otherwise. You don't have trials that are equivalent, that one is adaptive and one isn't, that you could get a direct comparison. We also wanted to investigate through qualitative research the reasons for differences and to provide guidance for stakeholders like people in clinical trials units on resources required for adaptive trials. So a subgroup of us developed 5 different scenarios that were all based on real trials, but we'd made some changes because it was felt a bit sensitive to use a real trial. We didn't want anyone to feel like their costs that they had been submitted for the original trial were being criticised at all, for example. So we made some changes so that comparison to the actual trial wouldn't be possible to do. But for each one, we considered a different type of adaptive design, ranging from a group sequential design, which not everyone considers adaptive, but in my mind is a good example of an early type of adaptive design, through to dose finding trials that use an adaptive approach, and multi-arm, multi-stage, umbrella trials, sample size reassessment. So each scenario had a non-adaptive version and an adaptive version, which we'd written as a PICO document to summarize the trial. And the adaptive version included at least one interim analysis, but some, such as the multi-arm, multi-stage, had more than one interim analysis. We invited clinical trials units in the UK to participate in this, and 10 CTUs agreed to participate. Unfortunately, by the time we got to the stage where we were sending out materials for them to doing, it was March 2020. And you might have found yourself that the workload in clinical trials units massively increased with the COVID situation. A lot of trials were being paused and such like. And so the number of actual costings we got was not as high as we'd have liked, but it's still very much comparable because each costing had the non-adaptive version and an adaptive version. So we have a paired comparison. So 7 CTUs provided at least one costing. Some did all of them, and some just one or a low number. So we asked people to do what they could in the time available. So here are the overall results, and we divided the costs into staffing costs and other costs. And the staff costs were divided into, maybe not perfectly, but divided into different disciplines that you need for supporting a clinical trial. So statistics, data management, trial management. And some things to point out is that the median increase in cost compared to the non-adaptive version was generally quite low, except for sample size re-estimation where it was high, but that was because the adaptation resulted in a longer length of project, and that generally results in the staff resource being longer. And as I'll mention on a subsequent slide, all of the CTs kind of return costs for the worst case scenario, so the maximum cost that would be required. So that's one thing to point out, that the median increase that was found wasn't very high, but you can see the range. The range is very interesting. It's very wide. So clearly there's not very strong consensus on how much extra resources are required for adaptive trials. but if you look at statistics, that was the highest increase. Data management also, although in one scenario it didn't increase, the median wasn't increased, but other scenarios it was higher than the total. And the trial management was generally less than the others, although you can still see there's quite a big range there. And other non-staff costs, which might include, randomization systems and clinical data management systems and such like, you can see were generally relatively low increase apart from the sample size re-estimation. Here's the figure. Here's a figure showing it for scenario 1. We do have this for every scenario, but you can see what it looks like. So each CTU, which is done anonymously, and we're only benchmarking by what 100 represents the median for the non-adaptive. So we've done everything we can to avoid anyone criticizing individual clinical trials units for the high costs that they returned. But you can see this is just kind of representing the non-adaptive versus the adaptive for each CTU. So it kind of gives you an idea of the variability we're seeing, which is very interesting. But the main thing we're looking at is the difference between non-adaptive and adaptive. I think I've said all of that already. So after that mock costing exercise, we did qualitative research with the staff who did the costings from each clinical trials unit, and all of the participants agreed to be interviewed, and there were trained qualitative researchers who did that. So I can't really go into too much detail on this, on all of the qualitative time, but I think it was interesting that where the trial design had a variable duration, all of the interviewees are costed for the maximum. So it wasn't so possible to put in the average expected cost you'd expect and then ask for more funding. At least with UK funders, you kind of have to give one number, and that's going to be the maximum that you could plausibly need. With statistics, the increase was due to the extra interim analysis effort needed and a more complex protocol and maybe more advanced statistical analysis and design needed. For data management, this was generally changes were because of the randomization system, the database, and for perhaps increased amounts of data cleaning required for the interim analysis to give a good result, to give a reliable result. Trial management, where it was increased, was generally the rationale was due to complexity of design for protocol amendments and to be involved in data cleaning as well. The variability that we saw, so apart from the sample size re-estimation, which was because of the increased project length, the variability was partly related to the complexity of the non-adaptive design and the complexity of the interim analyses. So if we go back to the table, for example, the umbrella study, the non-adaptive version of that is already quite complex. So that's why maybe even though the adaptive version is very complex, the median increase isn't as high as you might expect. There is strong variability between CTUs, and that might be explained by the different funding that they receive from the universities or from core funding, for example. Also possibly related to their experience. So some CTUs were very experienced with adaptive trials. Some had only maybe done 1 or 2, or I think there might have been 1 or 2 that hadn't done any before. And I think understanding how the uncertainty in the sample size that you get from the adaptive design is something we would need to look into more, because all of these costs represent the worst case scenario. So I really hope that this would be out by now. It was accepted about a month or 2 ago in BMC Medicine, and I really wanted to be able to give you a link to the paper. I imagine it will be out tomorrow just by my luck, but hopefully it will be available soon, and all of the results from the mock costing exercise and the qualitative research are available. And we're now working on finalising a guidance paper that will help people plan the resources that they need for adaptive trials compared to non-adaptive ones. And part of this is also thinking about what funders could do to encourage wider use of these more complex designs. So just to finish, and I did get through everything in 45 minutes, so there you go. Hopefully I gave enough detail, but happy to answer questions. But master protocols, I think, offer great opportunities for clinical trials, and I really am very interested in how we can use them in other areas. So I've got a big interest in the inflammatory diseases, as you might have seen in my examples. But we do need further research on how best to design them and analyze them to increase their efficiency and informativeness. And we need more consideration on which elements provide benefit in different situations and what additional resources are needed to support them. So still a lot to do. And yeah, anyone who's working in the area, there's a lot to do. So carry on. So I'm going to stop sharing there.
Perfect.
I'm happy to take questions.
Perfect, James. On time. I think that was a very nice overview showing the different aspects of the different designs, innovative or complex designs that are currently getting more and more popularity. Questions from the audience, either through the chat or raising hands. Matthias?
Yeah, thanks very much, James, for a really, really nice talk. Very interesting. I have a very practical question which relates to the COVID crisis. Could you comment, and perhaps Sven could also add, how these new approaches have actually sped up the evaluation of treatments or vaccines, and where have they failed? Because in my understanding, the recovery trial, which was sort of really a landmark in that context, was a traditional one, if I remember correctly, but I may be wrong. So I just wanted to put this a little bit into context of the COVID crisis.
Yeah, thank you. I think that's a really good point, because these platform trials have really been used very widespreadly in COVID. And the recovery trial actually was a platform trial testing repurposed treatments. So it had the multiple experimental arms at once. It had the adaptive design that allows dropping of treatments, and it has the adding in new arms as they become available. So I mean, it was very successful in terms of finding treatments very quickly that improved standard of care, like dexamethasone, for example. And I think, I mean, a lot of, so the Stampede example, which was perhaps the first platform trial I'm aware of, at least, you could see was 2006 until like still going now. So I think it was amazing how quickly recovery like set up and started recruiting patients. And part of that, I think, was just everything in the UK, all of the research support infrastructure just prioritized getting it set up, opening sites and such like. And that's clearly not something that can be extended to every other setting in the future. We can't prioritize everything. So I'd say that these platform trials have been really useful for evaluating repurposed therapies quickly and have had some big successes. I'm not that convinced about the adaptive part of it, like I said, because the recruitment was so quick compared to the endpoint. The endpoint's relatively short, so it might still be okay. And there is so much resource and effort being done to do interims quickly. So I might be wrong on that, but I think in my mind, the adaptive part might be the part that provided least benefit, but still might have provided some benefit. But having multiple arms, having the potential to add in new treatments was really useful. And I'm going to ask Sven if he could comment on vaccines, because I think he's much more in that than I
cannot respond too specific to that. I think the recovery is a good example for how, or a nice example how the English system works and how it can fit into such a system. I think the other example would be the WHO trial. There's lots to say about the trial itself. I'm not going into the detail, but again, there also, it's mainly the multi-arm aspect that was of interest for the WHO, for the Solidarity trial, and not so much the adaptive part. The vaccine trial that is currently running, that started about 10 days or so ago, has adaptive aspects in terms of new vaccines to be added. It's also a multi-arm trial where there's pre-planned possibilities to add vaccines basically on the go, and it has the feature that it's at the moment a superiority trial, so vaccine versus placebo, but once there is vaccines available in the countries where it is running, it can be switched to a non-inferiority part, so there is an additional aspect to it, and that's good.
So you
more than that, I
the
don't think there is
placebo
lots to say.
Sorry, the issue is that at sometimes you cannot carry on with the placebo arm.
Yes, exactly. That's the idea. Beyond those two very prominent treatment trials that completed, I'm not sure how much there was actually, I have to say.
Yeah, I know that was quite a few platform trials started in different areas that didn't really go anywhere and were stopped early. But so there's been some really successful platform trials and some that maybe got set up for various reasons but didn't actually
which might point to another aspect or another point, which is that if we think we can set up these large and complex trials within same system and approaches we always have used, basically, then they're as likely to fail as any others, or even more likely to fail because they will never make it. But that was just a side comment. Other questions?
Does that apply to the WHO trial as well? That the WHO platform trial didn't really go very far, did it? I'm asking that also because the Swiss National Science Foundation put in quite a bit of money, and we're not sure whether that was a good investment. Anyway, yeah.
But that raises an interesting point. How would you define that the platform trial fails? You know,
perhaps what Sven said, that if it is, and James, when it's done within a health system, it is more likely to succeed than when it's a multinational, complicated, bureaucratic machine like the WHO will likely set up. So I don't know whether you want to comment on this.
Is basically no longer recruiting patients? So I might have misunderstood, but I thought that one elegant element is that you just keep randomizing to whatever you think are the current arms you need to randomize to, whatever the decisional process is, what to drop and what to add. I don't touch that, but
yes, that would be the idea, that you have like in Stampede, like what James said. Huh? I think that is, if it's done and running as intended, then it would be like Stampede, huh? Ongoing.
Yeah, I think there's, I don't know if it applies to the Solidarity trial, but there's some kind of modular platform trials where you can be part of subset of the arms and not necessarily all of them. So maybe that type of thing might help with international bureaucracy and such like, because yeah, I think recovery worked really well because it was just hugely prioritized and it was all within one healthcare system.
Yeah, and the money was coming on.
Sorry?
Somehow it was funded heavily, I guess.
Exactly, yeah. There was people who funded it like really quickly and took a chance on it, and clearly that one paid off. Not all of the investments have paid off, as you might have implied, but yeah, we're talking about relatively small amounts of money compared to the costs of COVID as a whole. So it's
there is a question by Dick. Maybe
Oh,
you
about
can
training.
add, was training costs considered, I think,
Oh yeah, so
in the costing?
we had training as part of one of the things that CTUs might include. Yes, the answer is yes. I guess maybe training specifically in adaptive designs might not have been thought of. I don't know, but we gave quite a lot of flexibility to what costs that CTUs would return. So if they would include training costs normally, then we asked for them to return whatever costs were normal.
Okay. Other questions, comments, experiences?
Is that question how much training costs, or?
No, what Matthias said, Solidarity was funded.
I don't know what the total budget was of Solidarity, but many funders chipped in very quickly, including the SNSF. You know, and millions. So
I don't know. I agree. Funding is if it's not coming, it's killing the projects, as James said on some of his projects. And then just funding alone isn't making it a success.
Then I have another question for James, and that relates a little bit, is a comment to what you presented about the discrepancy analysis, or it came to my mind then, and that's the complexity, especially of the analysis at the end, is getting in a direction that is difficult to communicate. And I mean, we already have difficulties communicating what a p-value is, what we've using since a long time. Now we're getting into areas which are much more complex. What's the solution for that? And not for the different audiences. So for the funders, for the public, for the clinicians, what can we do about that?
Yeah, really good points about basket trials and the analysis being more complex. And I mean, if you look at the separate analysis, then I guess that's a bit easier to communicate. I'm not sure about pooling it all together, but probably that's quite easy to communicate. Talking about Bayesian discrepancy, Hellinger distances and stuff is maybe not so easy to communicate. But you do get something that looks like the separate analysis. So you get like a estimate of the most likely treatment effect, and you get a credible interval. Credible intervals are perhaps actually easier to explain than confidence intervals, whilst maintaining accuracy. Although, yeah, maybe that's not completely right, but I think they're probably easier to explain accurately than confidence intervals. So yeah, I see what you're saying about, do they need to understand everything that goes into, do people need to understand the method? Is it okay to be a black box giving an answer? Probably not. But a lot of, if we're talking about communicating results to clinical researchers, a lot of the analysis approaches we're using in trials, you know, missing data approaches and such, like a causal effect, you know, estimates, they're all pretty complex, aren't they? So I'm not sure it's unique to basket trials, but it's certainly an issue to how to communicate.
Okay, perfect. I think we are at the end. If there is no other burning question, I would like to thank James for this very nice overview that hopefully gives some ideas to others, maybe to use this or investigate or get in touch with James to investigate whether that could be something for their projects. And
they come to you, Sven.
or to us, yes. And yes, there's always good. The more complex, the more easier people come to us. This is always good. And I wish you all a very pleasant evening.
Thank you.
Thank you very much.
Opportunity.
Great talk. Thank you. Bye bye.
Thanks a lot.
Bye, everyone.